So structures of a bacteria, basically the anatomy and physiology of a bacteria. So similar to us, as we have structures, bacteria also have structures. They have the cell wall, the cell membrane, cytoplasm, nuclear material, capsule, flagella, pili, and spores, but these are not in all bacteria, okay? So I want us to go through each and every structure, which is very high yield. You should know the structure, the chemical composition, and the function of that structure, okay? So one thing is we are going to divide this lecture into gram-positive and gram-negative bacteria so that we see the differences. So this is a gram-positive bacteria. One thing you'll observe is you can see the cell membrane and then you can see the cell wall. It's obviously dyed purple so that you know that it is gram-positive. And then you can see the nucleoid, there's the flagellum. It's a very basic structure. But now if you look at the gram-negative bacteria, you'll see they've added, there's the pili, there's the capsule. So capsule can be in both gram-positive and gram-negative. Then we have the cell wall. We have the cell wall. And then we have the cell membrane. No, before that, we have the outer membrane, which is very specific for gram-negative bacteria. Okay. And then we have the cell inner membrane. Then we have some ribosomes, granule. Okay. So this is a picture of a gram-positive, very basic. It only has the cell membrane and the cell wall. But for gram-negative, what I want you to remember all the time is we have an extra outer membrane. We're going to see its function. Okay. So that is the take home I want you to have. Number one, gram-negative bacteria have an extra outer membrane and gram-positive bacteria don't have that. Okay. Then obviously, why is it stained purple? It's because it has a very thick peptidoglycan layer that you're going to see. And for the gram-negative, a very thin peptidoglycan layer. Okay. I want you to remember that. So cell wall. It is the outmost portion. As you can see here. I don't know if you can see it clearly. So for gram-positive bacteria, can you see it here? One thing I want you to observe in the cell wall of a gram-positive is this one. Don't know if it's clear. It's called lipoteichoic acid. This thing. Okay, that is very unique for gram-positive bacteria. It is only found in gram-positive bacteria. And then here's the peptidoglycan layer. As you can see here, it's very thick. And then here's the membrane, the cytoplasmic membrane. Then for gram-negative, what you're going to see is we have this is the outer membrane, which is absent in gram-positive. So we have the outer membrane, then we have the peptidoglycan layer. You can see how thin it is. Then we have the cytoplasmic membrane. So the order is gram-positive, we start with the cell wall, which the cell wall basically is what has the peptidoglycan layer. Okay. So the cell wall, then the cytoplasmic membrane. For gram-negative, we start with the outer membrane, then cell wall, then cytoplasmic membrane. Okay. This is another picture showing. So as you can see here, it only the membrane wall or the cell wall, then cytoplasmic membrane. But here we have the outer membrane first, then the cell wall, then the cytoplasmic membrane. So I want you to remember that order. Okay. Have you understood? So the cell wall, which is the peptidoglycan layer, a backbone of NAG and NAM. NAG stands for N-acetylglucosamine and NAM. NAG and NAM. NAM stands for N-acetylmuramic acid. Basically, you know, if I were to draw the cell wall, it looks like this. It is joined together, then another one, then another one. Okay, so you'll find that this is NAG, NAG, NAM, NAG, NAM, and they are cross-linked together. Okay. Okay. So this cross-linking, we call them cross bridges. So there's NAM, NAG, NAM, NAG, NAM, NAG, NAM, NAG, and that is what is going to make the peptidoglycan layer. So a set of identical tetrapeptide side chain attached to NAM, different components and binding modes in gram-positive and negative bacteria. So we're going to see. But basically, the take home here is the peptidoglycan layer is made up of NAG and NAM, and they're bound together by these cross bridges. A set of identical cross bridges only in gram-positive bacteria. Okay. Okay, that is the peptidoglycan layer. As you can see here, I don't know if you can see it clearly, but this is the order of which it looks. So this is in a gram-positive bacteria. This is in a gram-negative bacteria. And I told you, remember, another quite unique thing about gram-negative bacteria is the lipoprotein. So it's known as LPS, but you're going to see this. All I want you to remember is gram-positive bacteria, thick peptidoglycan, gram-negative bacteria, thin peptidoglycan. Okay. Special components of gram-positive wall. So as you can see, this peptidoglycan, how thick it is. And then we have this LPS. Sorry, lipoteichoic acid, not LPS. LPS is different. Lipoteichoic acid. Can you see them? They are projections. Okay. This is only found in gram-positive cell walls. And then gram-negative, as you can see here, we started by saying there is the outer membrane. So outer membrane is made up of two things. We have these things called porins. Can you see these porins? Okay. And then we have LPS. So it depends. There's some bacteria that have LPS. It's called lipopolysaccharide. And then the bacteria have LOS, lipo-oligosaccharide. Okay. But they're very quite unique for gram-negative bacteria. And then here's the peptidoglycan layer. And then here's the cytoplasmic membrane. Okay. Is that clear? So functions of the cell wall. Number one, maintaining the cell's characteristic shape. The rigid wall compensates for the flexibility of the phospholipid layer and keeps the cell from assuming a spherical shape. Okay. So if the bacteria is a rod, if it's a bacillus, sorry, if it's a rod, if it's a bacillus, like that, the shape will be maintained. Countering the effects of osmotic pressure. So if you remember what osmotic pressure is, osmosis is where fluids move from high concentration to low concentration. So it's going to counter that effect so that fluids don't move from maybe from the outside to the inside or the inside to the outside. Okay. Providing attachment sites for bacteriophages. Yes, we did bacteriophages in virology. These certain viruses that are used to administer, let's say, certain drugs. Okay. Then providing a rigid platform for surface appendages, flagellae, fimbriae, pili, all emanate from the wall and extend beyond it. Okay. Play an essential role in cell division, be the sites of major antigenic determinants of the cell surface, then resistance to antibiotics. Okay. So those are the functions of the cell wall. So the main thing I want, if you're asked about the composition of the cell wall, we have understood that it's made up of peptidoglycan layer that are cross-linked by the transpeptidase, okay? The NAG and NAM structure. That's the main thing that I want you to remember from the cell wall, okay? Now we go to cell membrane. So cell membrane, bearing the enzymes and carrier molecules that function in the biosynthesis of DNA, cell wall polymers, and membrane lipids. Electron transport and oxidative phosphorylation, excretion of hydrolytic exoenzymes. So, as you can see here, so the cell membrane, remember the order here, outer membrane, cell wall, and then cell membrane. So here we're talking about the cytoplasmic membrane. Okay. So basically it's a phospholipid layer, similar to the ones we have in our body, you know, hydrophobic and hydrophilic end. Okay. So cell membrane, what I want you to remember is it's just a phospholipid layer. And then we have enzymes here, okay? So there's a very important protein we are going to learn. It's called penicillin binding protein. So how to counter this bacteria? We have an antibiotic known as penicillin. I'm sure you've heard of it. You've heard maybe of amoxicillin, ampicillin. Those are the most common penicillins. So they usually, the site of action is usually in the cell membrane, okay? And then another thing I want you to remember is the lipoteichoic acid, which was from the gram-positive. Where is it? This one. You see it extends from the cell, the cytoplasmic membrane. Can you see it? Okay. And it is only found in gram-positive. So lipoteichoic gram-positive. So you'll remember with the P, lipoteichoic gram-positive. Okay. We're going to see the function, or if I was to mention, so lipoteichoic acids, the function in general is it's going to induce, we call it tumor necrosis factor alpha and interleukin-1. Okay, so once it induces this, is now there are going to be changes in the body after induction of that. Okay, so like tumor necrosis factor, you'll learn this in immunology. The main thing it causes is it causes fever. So you'll see people who are infected with bacteria, especially gram-positive, you'll see they have fevers. So that is because of the tumor necrosis factor and the interleukin-1. Okay. Fever and also sometimes hypotension. So hypotension is low blood pressure. Okay. That was just to mention, but we're going to get into that. They are called endotoxins. So are we clear with the cell membrane? Then we go to the cytoplasm. So from your basic biology, we know cytoplasm is the fluid that is found within the bacteria. So consists largely of water, some together with proteins, nucleic acid, lipids, and small amounts of sugars and salts. So we can have ribosomes, okay? Numerous, 15 to 20 nanometers in diameter with 70 S distributed throughout the cytoplasm, sensitive to streptomycin and erythromycin, site for protein synthesis. So streptomycin and erythromycin, these are antibiotics. They go bind at the ribosomes, so preventing protein synthesis. Okay. And then we have plasmids, so extrachromosomal genetic elements. Okay. So what I want you to remember is in bacterial genetics, we have the chromosomal DNA and the plasmids. The main thing about plasmids that I want you to remember is they are responsible for resistance. Okay? They are responsible for resistance. What does resistance mean? Resistance means that you'll hear many microbiologists, they'll tell you maybe this certain bacteria is resistant to a certain antibiotic. The function of an antibiotic, that drug, is to kill the bacteria, but you'll see that it is resistant, meaning that the bacteria has mutated and it cannot be killed by a certain antibiotic. So what is responsible for that is the plasmid. Okay, extra chromosomal, meaning they're outside the DNA. Okay, then inclusions, sources of stored energy, e.g. volutin. Okay, so I want you to remember plasmids responsible for bacterial resistance. Oh, sorry. So we're done with the cytoplasm. Then nucleus. So this is the organization of the chromosome. We have protein is the domain, then double-stranded DNA. So they lack a nuclear membrane and absence of nucleoli, hence known as nucleic material or nucleoid. Nucleoid means, if you hear the word oid, it means similar to. Well, that's why it's called a nucleoid. It's similar to a nucleoli with several nuclear material or nucleoid, 1 to several per bacterium. Okay. And then we have the capsule. So capsule, you'll hear the word encapsulated bacteria. So a capsule is found on the outside of the cell surface. Okay. When this is organized into a shapely defined structure, as in pneumococcus. Can you see this capsule? The one that is white. You remember I told you about, I'm seeing someone has a question. Agutu, yes, you can unmute.
Yes, could you please repeat there's something that you've explained about the nucleic and the nucleoid, the previous slide.
Yeah. Or as I was saying, if you hear the word oid somewhere, nucleoid, an oid means similar to something. As we have said, the bacteria, they don't have nucleoli, okay? This is their nucleus, but they don't have nucleoli or a nuclear membrane. So theirs is similar to the nucleoli that we have in human beings, okay? So it is known as nucleic material or a nucleoid, okay? I don't want you to get confused. But basically, what you remember is they have a chromosome. Just remember that, which is known as a nucleoid. So passing of genetic material. When bacteria want to multiply, they have to pass their genetic material from one bacteria to the next. So this is what they usually pass through. It's known as a nucleoid. It's not similar to ours, which is a nucleoli. Okay? Is that clear?
Yes, thank you.
Okay. So as I said, the capsule. So as I said, you'll hear the word encapsulated bacteria. Okay? So this is the capsule that we're talking about. So a capsule is mainly made up of polysaccharides. Okay? And the main function of the capsule is it is antigenic in nature, meaning it will protect the bacteria against phagocytosis. You know, phagocytosis, the process where macrophages come, they engulf the bacteria. So it will protect against that process, against adhesion, okay? And it also provides as a virulence factor. So virulence factor is where the bacteria is fighting, okay? It's fighting against the immune system. That is the function of the capsule. And then you'll see something known as slime layer. So this slime layer, it's made up of, it's a loose network of polysaccharides, okay? It's usually found on the outside of the capsule, loosely attached. So here, the main function of the slime layer, it's slimy in nature. So meaning it will adhere to surfaces, okay? You'll hear the word biofilm formation. Basically, these bacteria, they adhere to the surfaces of materials. For example, you have put a catheter. A catheter has been put on someone's, you know, who knows where catheters are put? Who knows what a catheter is? Catheter.
So it depends on what catheter we are referring to. It could be urinary catheter, it could be a central line catheter.
Mm-hmm.
Yes.
Okay.
So if it is urinary catheter, it can,
continue, sorry.
it can be urethral. I'm saying if it's urinary catheter, it can be urethral or suprapubic. If it is about hemodialysis, it could be maybe from the important blood vessels, say the subclavian or internal jugular.
Good. So a catheter is a tube that is inserted into the body. So there are usually two main functions. It will either remove the fluid or to drain the fluid or to deliver medicine. So in this case, it can be any of those. They're called indwelling catheters. So for example, let's use the urinary catheter, where it's usually put in the urethra. Maybe it is used to drain urine from the bladder. Now, this slime layer of certain bacteria is going to adhere to the catheters. Okay. Now, when that adheres to the catheter, it causes something called as, it forms something called a biofilm. Okay. Biofilm. A very bad bacteria that forms that is called Streptococcus epidermidis. It's very bad because it forms biofilms on indwelling catheters. So if you're asked which bacterial structure is responsible for that, it is the slime layer. Okay. Then we have flagella. Flagella, as you can see, these outpoachings, it has only one function, motility. The bacteria has to move. Okay. So there's these different types of flagella. I mean, I've seen, we have been taught, but it's very low yield. They won't ask you. Monotricate, where there's only one, amphitricate, lophotricate, peritricate. I've never seen anyone ask you this, but this is just maybe good to know. Where there are, as you can see in this bacteria, it's only one. You can see in this one, there are multiple. Okay, so they have different names. But I've said the function of flagella is just motility, and it's made up of proteins. Okay. So this is the flagella, as you can see. If you were to zoom in here, can you see this? As it extends and it goes to the outside. So this is the direction of rotation, as like this, as the bacteria is moving. Okay. And I said it's just made up of protein. Yeah, that's the most important thing you have to know about a flagella. And then we have pili. So pili, don't confuse with flagella. Okay. First of all, a pili, it's not made up of protein. This one is made up of glycoprotein. Okay, so there's some. So pili are hair-like projections from the cell. They are known to be receptors for certain bacterial viruses. Chemical nature is pili. Can you see these hair-like projections? Similar to villi. You know how the villi are in the small intestines, the projections. So this is similar. Okay. You'll see some call it pili, some will call it fimbriae. But the main difference between pili and fimbriae is just that a fimbriae or fimbriae are just smaller pili or shorter pili. Okay. Can you see them? So for this pili, what I want you to remember for the pili are two things. Number one is their uses. So common pili or fimbriae, the shorter pili, are fine, rigid, numerous, related to bacterial adhesion. So adhesion means to stick. So where is this relevant? In bacteria such as E. coli, which is the gram-negative bacteria we're going to do, is these pili, they attach to the urinary tract. Okay. So that is the function of the pili, the common pili. And then we have the sex pili. These are longer, coarser, only 1 to 4, related to bacterial conjugation. Okay. So this is, conjugation is basically, it's involved in bacterial genetics, where one sex pili moves from one bacteria to the next. Okay, this is found in bacterial genetics, but I want you to remember that there's the common pili, which is used in adhesion, and then sex pili used in bacterial genetics, passing of genetic material. Another thing I want you to remember that I forgot to add in my slides is there's a certain bacteria called Neisseria gonorrhea that causes gonorrhea. If they ask you what bacterial structure is involved in antigenic variation in Neisseria gonorrhea, the answer is pili. Okay? If they ask you what bacterial structure is involved in antigenic variation in Neisseria gonorrhea, the answer is pili. Are we clear? Okay. I'm sure we're going to see that, but I just had to mention this so that you don't forget. So 3 things to remember in pili: bacterial adhesions, for example, E. coli in the urinary tract, sex pili during bacterial conjugation, and Neisseria gonorrhea. The structure involved in antigenic variation is the pili. Antigenic variation, if I was to mention just a quick one, is where it's a process where these bacteria, they change their surface antigens, okay, to evade the immune system, okay? That is antigenic variation. You remember we did antigenic variation. Which virus did we do antigenic variation? If you can remember. Antigenic shift and drift.
Was it influenza?
Influenza, good.
It was influenza.
You remember, that's good. Endospores or spores. So these are the specialized structures. They're not found in all the bacteria. Okay. So it's a dormant cell resistant to adverse conditions, which are the high temperatures and organic solvents. Okay, and then one thing I want you to remember is spores are only found in gram-positive bacteria. Okay. So identification of bacteria, so antigenic, only the gram-positive bacteria. Pathogenesis and resistance. So their main function is to resist. So basically survival, if I was to say that. If I was to use one word to remember spores, survival, for the bacteria to survive. So that's why it's resistant to the high temperatures. So like heat, and then organic solvents like chemicals. Also dehydration. If you were to put it, if this bacteria was to survive in the sun, it would. Okay. Produced when starved, contain calcium dipicolinate, DPA, dipliconic acid, found in Bacillus and Clostridium. Okay, so this is not relevant. Okay. Just remember that they're used for survival. Spores, survival. And here they are. Can you see them? Usually found at the end of the bacteria. Some can be at the end, the middle, like that, like that. All best forms of bacteria, or the L forms. When bacteria are treated with, number 1, either enzymes that are lytic for the cell wall, e.g. lysoenzyme, or antibiotics that interfere with biosynthesis of petroglycan, wall-less bacteria are often produced. Usually these treatments generate non-viable organism. Basically what this means is when you use an antibiotic to kill the cell wall of a bacteria, these organisms die. Okay. We understood. And then we have the bacterial growth curve. This is also high yield. They can bring it in a spot where they show you this, and then you are to label what phase is this, what phase is this, what phase is this, what phase is this. Okay, so I want you to remember this. I want you to remember the order is usually lag. So as here, lag, log or exponential, stationary, and then phase of decline or death. So as you can see, lag, exponential, where they multiply, and then stationary, and then death. These are usually the growth phases of a bacteria. So if you are to see lag, basically here is where they're adapting to the nutrients. As you can see, the bacterial growth is not that high. Then we have exponential. Here is where we have the active growth. So here they're taking in nutrients, they're multiplying, and you see how the organisms are increasing. And then stationary, the growth rate is decreased. Basically, like if you are to put it in terms of a population, you can see here they're adapting to the, if you are to put it in human context, let's say, for example, 2 people go into a place, they're adapting to the place. Let's say you've removed someone from Nairobi, they're going to, let's say, Kwale County, they adapt to the place, and then they start reproducing, the generation increases, and then what happens here is the rate of reproduction becomes stationary, okay? Then death is when now they start growing, okay? So that is usually the growth phase of the bacteria. So death, it depends. Now here, maybe they have become too much, then nutrients, there are insufficient nutrients, or maybe an external factor has happened. So maybe you've used antibiotics, the pH has changed, okay? So things like that. So these are the phases of growth. Optimizes curves in production. Yeah, that is all I have in the phases of growth. Points to remember if we were to do a summary for the both lectures we have done. We have done the staining, fixation, cell wall of bacteria, the teichoic acid, lipoteichoic acid found in gram-positive, LPS found in gram-negative, and classification of all those cell wall structures, flagella, the spores, pili, and then the bacterial growth curve. Okay, and I'm done.